this Explain Everything is to discuss microarrays. And I just want to start off here by just indicating that this is a huge area. Okay, $4 billion industry just in microarrays. This whole field of biotechnology is huge. And if you were just to do a quick search on any of the techniques that we've been looking at, you can find out how much money is being invested in these various techniques and the success rate. That's something else I want you to appreciate, that just because we're looking at these experimental models for the gene therapy, for example, you have to look at where they are as far as clinical trials go, how successful they are, and in light of that, how much money is being made and how much money is being lost in this huge field of biotechnology. So there we go, $4 billion industry. When I initially did a search on microarrays, I came across this article. Now, it initially caught my attention because it is discussing morning people, which I am not. But then it caught my attention because it was talking about a 23andMe study. I'm not sure if you're familiar with this. It's been extensively advertised on television. So then I thought I would just go and do a little bit of research into this study, this 23andMe study. So here's the online advertising for this 23andMe kit. 199 Canadian dollars, and you can get information on over 100 health conditions and traits. Didn't realize how much controversy had arisen about this. Okay, in 2013, okay, the FDA shut down the sales. However, by 2015, they were back up. However, there's still controversy about this. Okay, they're talking about is it not only safe as far as medical advice, but what's happening to all the data that they are collecting from these. Okay, so there's articles about the fear of that. Okay, then there's other articles saying it's being overcautious. Then there's always the problem with perhaps who is going to access this information. So the purpose of this Explain Everything is not to get into any of these controversies. You can certainly do a search and find out all the information and make your own decision about this. But anyway, I'm going back now to the original article to talk about the microarray. So when looking at one of these microarray studies, what you have to determine is what are the two groups that they were looking at. Okay, so here again, we would be looking at morning versus evening, and then look and see, did they find any unique genes linked to either of these groups? And in this case, they found 15 loci. Okay, loci are just locations on the genes. So if we were doing a microarray, on the next slide I will show you how they would have determined that these were 15 unique gene expressions in the morning versus the evening people. So let's look at the procedure had a microarray been used to determine which genes are being expressed in the morning versus the evening. So first step is to harvest the mRNA from both of these groups. Okay, and then create cDNA for both of them. Again, recognizing that in microarrays you create the cDNA using the reverse transcriptase for both groups. Okay, reverse transcriptase, and then you tag this unknown complementary DNA with different fluorescent tags. So we're going to tag the cDNA from the morning, let's say with red, and then the cDNA from the evening with green. We then combine these and spread them onto the microarray slide. Now remembering that these are going to be known DNA. Okay, so all these little bits are known strands of DNA. And what we're going to do is then look at what binds. So for the morning, we would have some unique morning, and we might have some unique evening, and then we might have some ones that were common to both, in which case they would be yellow. Okay, so remember the common are yellow. Okay. And the ones that had nothing attached would not exist in either of these. We could then take the ones that you were unique to the morning, and they said that they found 15 loci. Now they're talking about the DNA. They didn't actually do a microarray for this, but we could have gone further with this. But let's say that they found 15 unique expressions, unique gene expressions for the known. Sorry, for the morning people. So they would take those, okay, take all those, and now create another slide with those 15 known, and that could act as a screening for morning people.